Okay, Brenda, shall I get started? You're good?
I'm good. Great.
Great. All right. Thanks, everybody. Thanks for joining us. A few more people will trickle in as we start, but I don't want to delay this. For ATP Bio Seminar Series, we are aware to have Professor Brenda Ogle from University of Minnesota today. Many of you know her already. She's a pioneer in the development of functional cardiac organoids. She's a faculty at Biomedical Engineering in University of Minnesota, the former department chair, as well as the director of Stem Cell Institute at UMN. At year, as we enter the second phase of ATP Bio, our goals are to develop actual manufacturing facilities, cryopress, and basically be able to develop the cold supply chain, cryo supply chain for all the biological systems we are working on. And one of the highest interest test beds that we wanted to get into is organoids. And Brenda was kind enough to agree to be our organoid test bed lead starting year 6 of ATP Bio. She is also a pioneer in actually manufacturing these very intriguing biological systems and providing them to researchers and clinicians. So we are very excited to work with that, and hopefully this conversation will start kindle even more collaborations between everybody. So without further ado, Brenda, thank you again for agreeing to do this, and the floor is yours.
Great. Thank you so much, Corkit. Thanks to ATP Bio for the platform to share some of the ideas that we have surrounding organoid manufacturing, preservation, and distribution. I thought I would start with a little bit about the lens that I come from, and that is cardiovascular disease. Some of the models we've been developing, and then how that has sort of evolved into the concept of an organoid manufacturing facility. And I'll say organoids in their truest sense is what we had initially envisioned. That is an aggregate of stem cells that you drive to become the organized cells of a particular tissue type. But we also work on microphysiological systems that can include microfluidic devices or our bioprinted or our gel cast. So this is going to be a mixture of all of that. So the long and short of it is that we've been working to develop these types of systems for a long time so that we can answer some of the exciting biological questions that we have in mind, and also so we can move towards treatment for cardiovascular disease. And along the way, we've also helped a lot of others try to use some of these systems in their own lab, and that's been also very rewarding and led to some wonderful collaborations. But I think we're at the point now where many other labs are who have these types of systems in their lab, and that the demand starts to outweigh the ability to produce them. And you start to have some inconsistency when you have a research grade product that you're moving between operators. And so the short version of this talk is that is what has led to me being very interested in exploring the concept of an organoid manufacturing facility. So let me start with some of our basic studies and some of the bigger contributions that we have made to the field and why we've ended up where we are. So we're very interested in the extracellular matrix. I teach a course on the extracellular matrix and how to control how the extracellular matrix remodels for engineered tissues, but also in vivo. And this schematic is from several years ago. Just going to move my little thing around so I can see all of the screen. And it showed some of the very many ways that the extracellular matrix impacts on cell behavior, well beyond the structural support that had been the dogma 30, 40, 50 years ago. And you can see all the things that happen. The ECM serving as a reservoir for growth factors, as a signaling trigger via integrins. And when those integrins or other cell surface protein are stimulated by the extracellular matrix, a range of intracellular signaling pathways are activated. And in 2014-16, these were some of the main triggers: extracellular matrix synthesis, so more remodeling of the ECM, invasion, proliferation and certainly adhesion. And my lab was interested in how stem cells specifically engage with the extracellular matrix. And what we found was that extracellular matrix proteins, and there are over 300 of them in your body, can drive the specification of stem cells. That was a big surprise for us then, and now it's just sort of very commonplace. And so what we've added is this sulfate process to the ECM-directed sulfate processes. So that sounds simple, but it really transformed the way we think about most of the things in our lab. And the first thing it meant is, okay, if particular ECM proteins are driving the specification of particular cell types of a given tissue, could we design an optimized ECM formulation to drive the specification of the cell types we're most interested in? And in my lab, they are cardiac cell types, especially cardiomyocytes, but including endothelial cells, smooth muscle cells, cardiac fibroblasts. The second thing we were wondering is, what are the signaling pathways that are stimulated to drive specification? So I'm going to share with you some of those outcomes just very briefly, again, to give you sort of a, I don't know, a picture of my background in lens and why we ended up where we did. So when we were thinking about engineering an ECM to drive the specification of cardiomyocytes, we looked at the extracellular matrix of native tissues. And at that time, there wasn't a lot in the literature, and there weren't still not very sophisticated methodologies for assessing the extracellular matrix. And so we went with, sorry about that. Immunohistochemistry, simple, for one representative ECM protein from a bunch of different families of ECM proteins. So we picked collagen, type 1 collagen to represent the fibrillar collagens, elastin, fibronectin as a small linking protein, and then type 4 collagen to represent the basement membrane. We tracked them over time, and you can see on the top panel, this is from an E12.5 embryo up to a P or postnatal day 2 embryo. And these are cross sections of the left and right ventricle. And so if you zoom in then here on the left, can you see my cursor? Yes, okay, great. Is the epicardium all the way through the myocardium to on the right is the endocardium. And at early time points for this particular extracellular matrix protein, you see that it's very sparsely distributed, and then over time it accumulates. But this is not the case for all extracellular matrix proteins in the heart. Actually, elastin is very prevalent at early time points, and then it sort of disappears and becomes most localized around the large vessels. And so we took that information, we quantified it as best we could, and we used it to set ranges for a design of experiments optimization. And for that, we used polyethylene glycol gels into which we entrapped different combinations and concentrations of representatives from those main families of ECM proteins in the heart. And Philip Jung did this work, and he developed not only the concept and the idea to use a design of experiments approach, but he also developed the polymer 3D approach. And this is just in wells, cast gels in a well. But it was really useful in being able to present ECM proteins to stem cells without covalently linking them to the actual polymer itself. And so the idea then is look at those concentration and combinations, feed it into the statistical model, come up with a theoretical optimum, which is here in the middle, and then test that optimum experimentally. Oops. Did I lose you all? Can you still see it?
We can see it. I see some black areas in the presentation.
What just happened? Tried to minimize the...
We want to stop sharing and try one more time. Sometimes it's a soft reset.
not getting my little thing to minimize. All I can see is you guys. I'm just going to leave and hop back into the call. I'll be right back.
Okay. Sorry about the intermission. There will be no intermittent entertainment while we wait for Brenda to join us. It should be momentary. Ellie, while we wait, I think we had a couple of announcements. Would you mind reminding me what we are supposed to do? So,
Oh yeah, it was just a quick reminder to let people know that we are recording today's webinar and it will be available on our YouTube channel as our past webinars. So if you've missed any of those, please visit our YouTube channel and you'll be able to play all the recordings that we've had from past webinars. And also just a quick announcement that we are moving into our spring series as of the new year. And the first one will be in February on the 17th, and it'll be the State of the Center. So watch out for the emails about that, and then please join us for that. And I think Dr. Ogle has rejoined.
thank you, Ellie. Looks good, Brenda.
There we go. Can you hear me now? Okay, great.
Yes.
So we were trying to come up with a formulation again that drove the specification of cardiomyocytes. And I shared with you the approach, and this is basically the outcome. We found that we could identify an optimized ECM formulation that drove the specification of cardiomyocytes, and that's basically shown in this lower left plot, where you see cardiac troponin T area. That's a protein of the sarcomeres for those not in the cardiovascular space. And you can see that the OPT, which is our optimized formulation, is approaching 50%, while our polyethylene glycol, that's our polymer only, is essentially nothing. And then if you maximize the concentration of those same ECM proteins, you do drive specification, but not at the same level as the OPT. That was transformative to us because now, without adding any other type of stimulation, we can drive cardiomyocyte specification. And this is important because unlike small molecules, extracellular matrix proteins can be spatially placed very easily without any type of carrier. And that means we can start to think about controlling the formation of tissues in a dish. And the thing that was also exciting about this result is that it was strikingly similar to the composition of embryonic day 16.5 embryos. So that just sort of validated our results, as did some work that another graduate student, Sophie Givens, did about 6 or 7 years later. Now we're moving into a better technology, mass spectrometry, to look at extracellular matrix profiles in a given tissue. She was looking in a completely different study for a completely different reason. The extracellular matrix proteins that were secreted by adult and fetal cardiac fibroblasts. So that's designated A, CFB, and FCFB. When she compared those, the ECM proteins that were expressed more in fetal cardiomyocyte or fetal fibroblasts rather than cardiac fibroblasts. And again, the fibroblasts are the main secretors of the extracellular matrix. She found the same main proteins that we found from our optimization. So fibronectin, type 1 and type 3 collagens, and laminins 211 and 111. So now we're pretty excited. We've got a formulation. And so we did this for a couple of other cell types in the heart. And I'll just show you, since this is a little bit newer work, the endothelial guided specification, because I think no matter what tissue type you might be interested in, they all need to be vascularized in some way, shape, or form. So this is a work from Michaela Hall, who looked at first just individual ECM proteins, not even in the context of an optimization. And what she found is that the components of the basement membrane that are most essential for blood vessel formation, 411 and 511 laminins, those both were able to drive the specification of endothelium. And here we're marking it with CD31 staining, and then we quantified that in the upper right-hand corner. And you can see that if we couple that with small molecule Wnt agonists, which are known to drive endothelial specification, we get even more. But even without that, 411 can drive specification. And that's kind of cool because that is the isoform that's present in the developing heart. So once she kind of was convinced that, yes, ECM proteins can drive the specification of endothelial cells, and kind of coolly, even better than Matrigel gel, which has been the standard that people are trying to get away from because it has a lot of lot-to-lot variability. 411 and 511 are really good candidates for this. So I wanted to take just a little dip into some of the work that we've done to investigate signaling pathways so that you can kind of understand, well, that sounds interesting, but how does it actually work inside the cell? And so she took a look in the literature and looked at ways that laminins engage with integrins and or how they can work to change the expression of other types of ligands that can basically feed into pathways that we know are responsible for specification. So I'll just talk about engagement with integrins. So when engaged with alpha-5 beta-1 or alpha-V beta-3 integrin, laminin 411 triggers the phosphorylation of several components of the focal adhesion complex, including integrin-linked kinase and focal adhesion kinase. And integrin-linked kinase is especially exciting because it blocks the phosphorylation of GSK3 beta, which is downstream of Wnt. And as soon as that complex is degraded, beta-catenin can go into the nucleus and control transcription of several arterial and mesoderm-related genes. And so if you then look at some small molecule inhibitors of FAC and ILK, namely CPD22 and PF5732228, you can see that that initial specification that was driven in both the control and DMSO, if we look at type 1 collagen exposure compared to laminin 411, you see that in the control of DMSO conditions, laminin 411 is great, drives specification. When you add the inhibitors, it almost completely blocks that activity. It's not until you fully block with IWP2 that you see a complete loss, but certainly these kinases are involved. And we went a step further and utilized a line that Michael Kaiba had that we were able to use, which was a DOX-inducible beta-catenin IBS line that basically allowed for beta-catenin to the transit into the nucleus in the presence of DOX, regardless of whether or not Wnt was active or the GSK3 beta complex was intact. And so if we look at collagen in this lower right-hand plot, in the absence of DOX, you can see we're still at our low level. Beta-catenin is not accessible, and collagen is the only ECM stimulus. Then we move into 411 minus DOX, and we see that characteristic increase that we had seen in the other plots. Now here's where it gets interesting. And then if you add DOX to the collagen condition, and you now have access to free beta-catenin, up it goes, up to the same level as the 411. And even more interesting, the 411 doesn't go up any further. So there isn't anything additive, which basically essentially keeps pointing us back to we're activating, we're allowing, somehow triggering beta-catenin to enter into the nucleus, likely through this altered Wnt pathway. So that was very exciting to us because now not only do we know that the ECM is a potent stimulator, we're also starting to understand how it's driving specification. And those studies continue on in our lab. She went further on and not just looked at single ECM components, she also looked to see if we could have multi-component interactions that would better drive or improve our efficiency of specification. And with this, we're looking at CD31 as the outcome. So looking for high CD31 expression. These are just a myriad of plots to show you the first half of the factorial experiment and 2 component interactions. So collagen 1 with type 4 collagen with laminin 111, laminin 411. And you can check out the paper if you're interested in looking closely at those contour plots. This is the first just outcome of the straight response surface quantification, and it's a lot to look at just to expose you so that if you want to look in more detail at how we approach this. And essentially then, once you have the response surface, you can back out an optimized formulation of those components based on the entire parameter space, just like we did for the cardiomyocytes. And we came up with a formulation that contained both collagens type 1,4, laminin 411, and fibronectin. Interestingly, we included fibronectin not because it's present in the basement membrane, because it's not, or it is at very low levels, but because for human-induced pluripotent stem cells, it was the only thing other than Matrigel that we could use to secure the iPS cells to the dish while we were running the assay. And so, I mean, that speaks to a lot of things. One is the requirement of ECM for iPS attachment, but it was really kind of a practical Band-Aid that once we had the combination, we actually didn't need fibronectin anymore. And when we removed fibronectin, we got this huge increase in CD31 expression. So fibronectin was essential for survival of the iPS cells, but was blocking in a lot of ways the CD31 expression. So that was an exciting result. This is also just to show that at the optimized levels, we see a much higher CD31 efficiency or expression efficiency than at maximum levels of those same ECM proteins. Again, just pointing to, it's not just throw a lot at them, it's throw the right combination and concentration, which is what obviously the cells are doing in vivo. We took it one step further and also just added in some of the standard components that are known to drive endothelial specification, including VEGF, vascular endothelial growth factor, and added another layer of ECM on top of these 2D monolayers that were on a ECM coating, essentially. We also added SB, which is a TGF-beta inhibitor, and that basically stopped the contraction or contractile capabilities of fibroblasts that were contaminating cells in the culture. And so you start to get this cobblestone morphology instead of the vessel-like morphology that we see without SB, which is another, I would say, as an engineer, another lever that we can start to manipulate and pull as we're trying to not only drive specification, but organization of endothelial cells in engineered tissues. Now for the application. So these formulations, what we were calling them at the time, really easily merged into the field or application of 3D bioprinting. So we know this as 3D printing, but with biologics. And for us, the biologics are exclusively proteins and cells. And so what we were trying to do here is to see if we could effectively 3D bioprint this endothelial-specific ECM formulation. And if in printing them with stem cells, if we drove exclusively the specification of endothelium, or at least enriched for endothelium using that ink. And so on the left, you do see that the optimized formulation. The second one in from left is the cardiomyocyte optimized formulation, and you could see here very few little pink dots, so not many endothelial cells differentiated in that context. And then we added more fibronectin, knowing that it would inhibit or should inhibit endothelial specification, and indeed it did. Still saw a lot of cells, but very few of them specified to endothelium. And then on the right, no extracellular matrix control. That just means we are left with our base material that we use for 3D printing, and that's gelatin methacrylate. And so you might go, oh, interesting. I see some endothelial cells there and they didn't use their optimized bioink. That's true. And we are trying to sort of uncover that as well. We still see more in our optimized ink, but we do still see them in the gelatin methacrylate only. And if you're in this field, you know that it's not just the biochemical signals that the ECM provides, it's also the biomechanical cues. And we think there's something related to stiffness here that also can help guide specification. So this is a grant figure that I like to show because it was a hypothesis and sort of a dream that we had many years ago that we would be able to have an ink that can, if we printed with stem cells, it would drive the specification of cardiomyocytes, and then an ink that would drive the specification of endothelial cells. And if we position them correctly spatially, because of that advantage I talked about in the beginning of the ability to pattern extracellular matrix proteins because they're insoluble, that we could then really organize a tissue, sort of a forced organoid production. And this is what it looked like. So if we print the vessels with it, vessel-specific ink and the myocardium with the myocardium-specific ink. And it's interesting because that dream is starting to become something of a reality. So this is a paper we published a few years ago now using the cardiomyocyte-specific ink. And again, the concept was to print with stem cells, and that's the panel you see on the left, and the little yellow dots. And then we allow them to proliferate, to take up space, and then drive the specification. And then by about 24, 25 days, they're starting to mature in this context. And you might be thinking, what is the deal with that geometry? I mean, it sort of roughly approximates a heart, maybe, but what we were trying to mimic is a ventricle, and a ventricle that we could perfuse. So that's why you see these two open ports here on either side of this basically open sac. We want to do that for a couple of reasons. There didn't exist a model wherein we could not only measure pressure volume dynamics like you can in a clinical case, but also impose different pressure volume mechanics corresponding to the development of different disease states. And also, secondarily, we were interested in developing something that was large enough to be used by our local medical device industry to evaluate medical devices in the context of living human cells. And I don't have the, not sharing slides of that, but we have used engineered tissues for just that purpose. We published something on using it to evaluate cardiac tissue after ablation. And we're continuing on with several more studies in that vein. So just to show you a little bit about this model, we call it the human chambered muscle pump, or HChamp. It can be perfused, as I said, if you connect tubing, essentially. That's what this video is showing. On the bottom panel, you see the print template and then the actual print itself, an overlay of the template plus print, so you can see just how good is the fidelity, and then an image of the actual structure. And again, it's about 1.5 centimeters on its longest axis. This slide just shows the composition of cells in the HChamp. Along the top, you see we looked for cardiac troponin I, which is indicative of cardiomyocytes, smooth muscle actin, indicative of smooth muscle cells, and CD31, indicative of endothelial cells. And you can see that the vast majority of the cells are cardiomyocytes, which is what we intended at the time. We hadn't yet added in our CD31 or endothelial cell specific ink. You can also see that because we allowed it to expand over time, we get a pretty nice contiguous muscle layer on the outside, and that is what is enabling the beating that you see on the left. This is spontaneous activity, and it is actually capable of moving small amounts of liquid through this quasi-pump. Then on the right, you just see staining for another sarcomeric protein, cardiac troponin T. And if you zoom in, you can see some of the sarcomeres. This is just some further characterization that was exciting for us at the time because we found that the cardiomyocytes in this structure and the way that it was beating enabled maturation of the cardiomyocytes in the structure. So to this point in our 2D cultures and even some of our other 3D models, we haven't seen expression of some later markers found in late neonatal cardiomyocytes of human, namely ryanodine receptor, BIN1 associated with the T tubules, inward rectifying potassium channel 2.1, and SERCA2. So just for those who are on the call who might know about maturation and how challenging it is sometimes to drive the complete maturation of a stem cell derived cardiomyocyte, might appreciate that, or stem cell derived whatever other cell type you might be interested in. So then we did some electrochemical characterization. This is going to show you some a voltage sensitive dye. Now we use that in Lena Telkachova's lab to look at things like activation time or action potential duration. And so on the top here, I'm showing you sort of one of our first H-champs. And when you see the flash kind of go across it, you can kind of make out the outline of the H-champ. And you can see that you have fairly contiguous muscle function. If we look here on another example, we also can see that. But you can also see if you look at the maps that there are some black regions. And we were kind of concerned about that because our histology showed an intact muscle layer. What we found is that in most cases, there were cardiomyocytes there, and we could see them if we then use point stimulation to artificially depolarize the cells, and then we can get the whole thing to light up. So that was exciting, and that's the real advance, is contiguous muscle function that allows for pump function. And we can measure that. We do this in Dwayne Townsend's lab here at the University of Minnesota using a pressure transducer and a conductance catheter to get volume measures. You can see the pressure volume graphs here on the top and the associated pressure volume loops that look almost nothing like the clinical case because we don't have valves at this point. But you can see the change with exposure to a drug like isoproterenol. And now we are incorporating that H-champ into a bioreactor so that we can add valves to the external portion of the flow loop to essentially mimic a native cardiac cycle. And this just sort of shows you what the quasi sort of mitral and aortic valve would do in terms of opening and closing to drive that loop structure, and also synchronized with a controlled beat of our H-champ and not just the spontaneous beating that we see at early stages, at least. Spontaneous activity is characteristic of embryonic and fetal maturation state. This is just a little zoom in of our current bioreactor, just to give you a sense again for scale. This is our current version of the H-champ and how it looks when it's plumbed into the reactor. And we can back out not only like flow profiles from beads, we've also done 40 flow MRI and some COMSOL simulations of flow to get a sense for velocity, pressure, and stress, all shear stress on the H-champ. So again, we can model native healthy conditions and also pathologic conditions. So that's kind of the model system, one of the model systems we use. And then, you know, as an academic, we're always interested in like making that model better, sweeter, you know, like adding more functionality. And for us, one of the things about the H-champ is it's robust, but it also is quite thin and could utilize or would benefit from a more robust remodeled extracellular matrix. And the main remodeler in the heart is the cardiac fibroblasts, and those are largely derived from the epicardium. And so that is what one of the functionalities we have recently added and published. Just quick epicardial biology. The epicardium is that single layer that surrounds the heart, but some of the epicardial cells undergo endothelial to or epithelial to mesenchymal transition to form the blood vessels inside the heart and also the cardiac fibroblasts. So the other kind of bonus to having a more robust ECM structure is that epicardial cells and epicardial-derived fibroblasts and smooth muscle can promote really essential characteristics of cardiomyocytes. And so epicardial cells can drive proliferation of cardiomyocytes, but they also start to drive electrochemical maturation, and that's what Sophie Gibbons showed recently. So this is just to show you the kind of timeline of how we, in this case, it was just a simple co-culture, not yet in the HChamp. She did some single cell RNA sequencing, and I won't go into a lot of the detail here, but just to show you that if you look at the population that corresponds to ventricular cardiomyocytes, it's this dark blue. The conditions are cardiomyocytes plus a TGF-beta inhibitor, cardiomyocytes, and then cardiomyocytes plus epicardial-derived cells and epicardium. You see a very distinct shift of this population from this sort of one lobule to this other. That means they're still cardiomyocytes, but they have undergone a significant phenotypic shift just from the co-culture with the epicardium. And she went on to show that the majority of this corresponds to the ability to proliferate, so more proliferation with the EPCs and the epicardial-derived cells, more maturation of the sarcomeres in terms of sarcomere length, and again, as I mentioned before, electrochemical function. So this is calcium handling, and we're comparing using these metrics, time to peak and maximum amplitude. And you can see a statistically significant improvement with epicardial cells and epicardial-derived cells. In the interest of time, I'll skip this because I want to get to the next part. So I'll just end by saying what I mentioned before is as academics, we're trying to always improve these engineered model systems. And we continue to do that, trying to integrate an epicardium, an endocardium. We're using it to study cardiac disease states, especially those that are monogenic, and so easy to make manipulations to create a stem cell population that has that particular genetic difference. And so we're looking a lot at myosin heavy chain mutations that drive hypertrophic cardiomyopathy. We're also thinking about this in the context of a rodent model to see if the pump can actually pump in the context of the vasculature of a rat. And then finally, as I already mentioned, we are using it for evaluating medical device interventions and especially following living cardiac cell types over time following the intervention, which is something we haven't been able to do. So while we're trying to add all these bells and whistles, meanwhile, there are a lot of other labs who would like to utilize not just this model system, which we call an organoid because it's derived from stem cells that proliferate and then differentiate. There are lots of other labs like ours that make model systems and want to make them more accessible or alleviate lab distribution. This is just a very small sampling of a few universities that have stood up, like we have, sort of an organoid core facility that can help distribute organoids or microphysiological systems. The issue is really scaling and distribution. And while we were kind of thinking and working through this at the University of Minnesota, we started talking to our partners at Mayo, and they're having the same issue. How do we improve access of our model systems? And that led us, largely at Mayo, their model systems, they're using patient-derived cells, and we're using a lot of stem cell-derived cells. So we thought we could kind of partner to think about ways to drive production and improve access. And this concept was funded by a state program called the Minnesota Partnership. And it got us thinking about how we could really move toward products and model systems that people can use more broadly. And that sort of morphed into a concept that was shared in a perspective paper recently that I'm showing here from a really talented group of investigators who have been thinking about this as well, and who kind of represent different aspects of what would be sort of the workflow if we were to scale production, drive quality control, high quality control metrics, preserve, and then distribute organoids. So you can check that out, and I'll sort of expand on that idea here. Kind of got together and we're thinking about what are the major obstacles that limit the ability of lab-grown organoids to make research impact, just broad strokes. And the main obstacles are inconsistent starting material. And, that's near and dear to my heart, given the extracellular matrix work that we've been doing. And I know the pains of inconsistent starting material. So that's one. Second is limited ability to generate large batches. And the third is inconsistent evaluative metrics. There are several entities, including SCB and NIST, who have started to define quality metrics for organoids, but it's largely been inconsistent and somewhat slow in sort of becoming established in the field. And then limited ability to share and store is probably the biggest. And so you can see that to address those obstacles, we started to think of, well, the simple black arrows, right? Like, how do we validate starting material? How do we standardize quality metrics? How do we accelerate and scale production? What role do bioreactors play? And then what role does cryopreservation or any kind of preservation and distribution play? So that we can realize on the right this sort of incomplete table of potential impacts that these model systems might have. And in the meantime, while these ideas were kind of hatching over the last 5-6 years, in this last year, it's become very poignant at the federal level because we have the FDA introducing a few years ago now the Modernization Act 2.0 to try to move toward human testing using human model systems to complement or fully replace animal testing. And then more recent directives from the NIH, further language from the FDA about, again, moving away from or supplementing animal testing. So in the context of this, it's become a national priority, not just an Ogle lab priority. And so we've continued to try to push these ideas forward. And that's part of the reason I'm excited to share this with you today, is because we don't have an answer yet. We have some ideas and some momentum, but we're really looking to find the best solution or solutions nationally. So one of the key pieces that I thought about in particular and was like perfect, that ATP-Bio exists and has been doing so well for the last several years. We started to collaborate with John Bishop's group, and Bhairab Singh has really been leading the way, another investigator here at the University of Minnesota, of trying to improve our ability to preserve cardiac tissue, which is, I think, one of the more challenging tissues to preserve for reasons that aren't fully understood. And we began to investigate the use of vitrification and the cryo mesh, which has been developed in John's lab, to try to enhance heat transfer. And we've been utilizing it not in the context of the HChamp yet, but in the context of another model system that is utilized quite a bit in our lab and is familiar and utilized in slightly different forms in other labs across the country and world. And that is engineered heart tissue, shown here in this upper right-hand image. And essentially a cast hydrogel, typically fibrin or collagen, where we can utilize our optimized ECM as well, seeded with cardiomyocytes and usually a mesenchymal cell type, like a fibroblast. And the reason that mesenchymal cell type is there is, one, it's good, it's sort of better mimic cardiac tissue, but really what it's there for is to contract the gel to bring the cardiomyocytes into close enough apposition to beat synchronously and contiguously. We couldn't do this with our HChamp because it would have compacted into a solid ball. But this strategy works for these little tissue strips. And they have been used very extensively for drug testing, but could also be used for medical device testing. And then just a reference for you if you want to check out all the other many different types of engineered cardiac tissues that are out there. So I'm just going to show you kind of a sneak peek at some success that we've had using this approach. And this is just sort of continue to fuel my fire for a manufacturing facility, because now there are organoids that can be cryopreserved or at least preserved for a certain period of time pretty well. But I think there's a lot of room for optimization and improvement, not just for cardiac tissue. So what I'm going to show you here are some movies that are basically the edge of an engineered heart tissue. And the flashing green that you see corresponds to calcium transient, so the movement of calcium or electrochemical function. And this untreated control, you can see it's nice contiguous green and it's beating nicely. Next, we have our cryoprotective agent handling control. So that just means it was exposed both to the CPA and it was taken off of the pillars and onto the mesh and back. What you can see here is there's a little bit of disruption, a little bit of speckling here, so maybe not fully contiguous, but the tissue still can beat at the macro scale level. And then we look at our cryo, and we actually see something that looks somewhat similar to the CPA handling control, with some added damage. But this is, to our knowledge, the first recovery of contiguous muscle function in an engineered heart tissue. And so we're really buoyed by these results, excited to continue to optimize this approach so that we can not only scale up production of an engineered heart tissue or an HChamp, but also store and then more easily distribute these tissues. So with that in mind, we started to, vision a little bit further. And basically by we, I mean the team, largely those who authored that perspectives piece, thought about what it would look like if organoids would come into the facility. And our vision is that probably those organoids would come in at a fairly mature state, meaning they'd been made pretty reliably in their respective research labs. We would bring them in and make sure that we had the best preservation approach for each of those, and then also store them at the facility and then distribute them. And that outcomes from that would be the same or similar to what I showed in the table before, applications in all kinds of things, disease mechanisms, drug discovery, toxicology, even adaptive computing. You could check out some of those references. They're kind of fun to read, and ecology, and regenerative medicine, of course. And here's a little bit of the workflow that we envision and some of the industry insights and partnering that we've started to bring together to be able to start with a series of 4 different organoid types: droplet-based, gel cast, microfluidics, and bioprinting. Starting really with solid, well-characterized starting material, addressing all those limitations, moving on to automated liquid handling, bioreactors, using AI wherever we can, especially with QC and refined workflows, and then working with standardization organizations to be able to define QC or QA, and then and not just at the endpoint, but along the way. And then ATP-Bio, of course, being a huge partner in this with preservation technologies. And then we have a molecular and cellular therapeutics center here on campus that has already been making and distributing cell products and biologics, even iPS-derived cell products, for several years. We know, though, that there's not though, but we know and are excited about lots of other things happening in the field. And I think I'm here today to sort of say, I think that we need a facility like this. I hope somehow with national agencies, partnering with industry, partnering with pharma, that we can come together to build at least one facility and perhaps many across the country, partnering with new and existing federal and industrial entities in adjacent spaces. For example, there was recently introduction of a standardized organoid modeling center that is just being stood up, but they're going to focus really heavily on data repository formation and also quality control and the utilization of AI in that space. And so there's a possibility of interfacing an actual standalone facility with this center. I also have been alluding to NIST and SCB for all the work they've already done in describing quality control metrics. And so, infusing those into such a facility. Thinking about ATCC and their model that they've used for so many years, I think since the 1920s, for collecting and distributing cell lines. I think that model could be very useful, and we could use something similar in this context. So I'm going to just end with that idea. Thank my lab, who were the ones who generated the data that you saw earlier, but then just really stop by saying, there are a lot of adjacent activities going on right now. And I think there's a real need to harmonize, synergize, and talk to each other. I know this is for the folks who are on this webinar, you might be very heavily focused on preservation. That will be a major part of this. But for others who are maybe on the other side of organoid development, I'd also be interested to hear your ideas. So with that, again, thanks to Corkit, you and ATP-Bio for the opportunity.
Thank you very much, Brenda. Wonderful talk as always.
Thank you.
We are certainly very excited to talk with you and collaborate more. We agree that these things need to come to like much more, manufacturing sharing level at this point. At this point, I'll just open the floor for questions. Just raise your hand, and I see Eric's hand up already.
Brenda, an excellent question. Sort of more of a selfish nature than to help your research, but to help our research more than that. What we see occasionally when we cryopreserve organs is we see the endothelium is preserved, and the CD31 is present, but the CD31 is diminished in staining intensity when we do confocal microscopy. So it may be just an opening to sort of say, are there suggested mitigation factors to help preserve either protein health or endothelial health after recovery? And of course, this goes across all of our organs and all of our institutions. So if you have advice from your experience, that'd be really, really helpful.
Yeah, I think so. CD31, PCAM, right? Cell adhesion molecule. And so it's maybe not surprising that it's damaged, right? It's facing the extracellular space. And so I think though we're trying, I'm not in the preservation space, but it seems like a lot of effort is toward preserving the cell and not preserving the extracellular space. And so I think, I am very interested in understanding which, how extracellular matrix proteins and everything in the extracellular space experiences cryopreservation. And if there are certain proteins that are less susceptible, certain that are more susceptible, and could we start to get to something where we use either like accelerated evolution approaches or something to come up with the most resistant extracellular space for a tissue? That will only help an engineered tissue, but maybe we can also know that, how we can or why we can best preserve certain tissue types. I also think there's something to be said for protecting the most essential functional components of a given cell. So for us, definitely the sarcomeres we see a lot of disorganization after freezing. And if we could somehow stabilize them, we've got a lot of kind of ideas in mind for stabilizing both the sarcomere proteins and also the ion channel proteins, because those are all the most critical components for function. Cell can kind of regroup with the rest of them, but I think they'll regroup much more quickly if they don't have to remake the sarcomeres, and maybe won't ever do that if the ion channels aren't working. So you're not getting good influx and removal of ions with each beat. You might just not recover function, and that cell might die. So
You bring up a good point, because we might, we look at a continuum from cells to organisms to measure health after a cryoprotectant or vitrification or high or low, high sub-zero. But maybe we need to step back one step even further and say what's common about proteins that are affected by these treatments, and is there something we can do to change the system to protect the proteins at the get-go?
right,
Thank you.
right. And I think, yeah, like ties in with the and that like ribosome display and other things where we can drive the best extracellular space would be really cool. So
Great.
Nathan?
Nathan?
Sure. Thanks, Brenda, for the presentation today. I'm just curious. Obviously, cryopreservation is a huge bottleneck to what you're trying to do here. I'm curious, what are the other major things that you feel that would need to be overcome in order to make this vision a reality?
I think the biggest thing for me is if people don't want to use these organoids because they don't trust them, they don't trust their quality. I don't think scale up and production are going to be an issue, and I don't think distributing them are going to be an issue if we can cryopreserve. But there is especially if they're going to start to like help with FDA submissions, people have to know that they can reliably count on these test beds and that they're going to be the same every single time they get them. And so I think that's a big issue. And I know the standards are kind of out there and they're sitting there, but I think that the thing that a facility could bring is just the robust manufacture and development of quality control, not just for like a bunch of different organoids, but for a particular organoid for a particular application. And I think once we have that kind of line and workflow, I think that's really good, what's going to drive the use of these model systems most effectively. They've already been used effectively for drug testing for sure. But to me, it's that piece. It's the establishing the quality, establishing its ability to predict a human outcome for a particular case or a panel of cases to start.
Thank you.
I had one question in the chat there from Raphael. I don't know if Raphael, you want that question still asked and answered.
I could ask it, sure. So I come from a mechanical engineering background, and in terms of 3D printing and making that accessible, obviously you need the 3D printer, but once you have that, if you have design files publicly available, then you can print whatever you want in the comfort of your own home or lab. Do you see like a kind of open source, like here's how you print an organoid and I can provide you the inks and here's like the CAD or here's like the 3D design files? Is that like almost like a decent? I mean, you're pushing for centralization. Do you think that decentralization has any what's it called? Any place in this space in solving this problem or no?
I think possibly. I haven't really explored that side of it, to be honest. I think that, yeah, I'm interested in like clamping it down, like bringing people in who've already really well established their workflow. Sure, most of these things are already available and accessible. It's just like the implementation in labs is not as simple as sort of, what would I say, like you publish a chemical formula and a lot of chemists or chemical engineers can make it, and it's the same thing every time, or mostly the same thing, right? It's so different when you're working with cells because they have, there are so many cues and signals that we don't yet understand. And that's what makes them highly variable. And so that's why I want to bring them together right now. I think it could be an offshoot, later especially as we develop them, it's like, okay, go ahead and try to make it, being in an academic institution, that's sort of my flavor. But I do think that how it's actually going to work is probably similar to ATCC, where somebody's got a licensed thing already, right? Like I've got a patent on my particular organoid or microphysiological system, like everybody does, right? And so if it came into the facility, we'd have to work out, what are the terms of the licensing agreement? What's the kickback on any revenue? And then go from there. So, but I
Thank
think it's
you.
yeah.
Yeah. It was excellent talk, and the work that you presented was really, really just mind-blowing, honestly.
Oh, thank you very much. I'm not sure.
Um,
Hey, Brenda, excellent talk. I have a quick question. I know we're getting close to the hour. You mentioned the different channels and sarcomeres, not losing them or maintaining them. Is there one, say, voltage-gated sodium or calcium that you think would be like the red flag or the domino that falls first, or even sarcomere proteins? Is there anything we could look at to see? I'm a channel person, so I'm a little biased what I would pick, but just curious in your opinion.
Yeah, I mean, I'm not really a channel person, but I think L-type calcium channel. What did you think? Yes?
That's my favorite calcium. I studied
Yeah.
voltage-gated calcium
okay.
channels, so you won my heart right there, no question. But they're amazing in the pacemaking, whatever. But
Right.
I think that
Right.
could be a key one for what it does for the heart in particular.
Yeah. And the one thing I haven't studied is just like how varied the structure is of the ion channels, which would be fun to talk to you about.
Yeah.
In terms of could there be like is it feasible to have a global protector of ion channels? Or, because the one thing we thought about with like sarcomeres, that isn't completely new, but if you just stop them from beating during the preservation process, you preserve them much better than if they're dynamic when they're being frozen. So could there be something like that for ion channels where you wouldn't kill the cell if you did that?
Yep. That's a great idea. The other one is the gap junctions that connects some of the cells as well. Could be another interesting one as well. No, I'll be happy to follow up. That's very interesting, great talk. And I think knowing that is like, could be prevent that first domino from falling kind of thing, as you just highlighted kind of thing. Maybe that would be what we need to help from rebuilding things. So let me think about that a little bit. It was a great talk. Thank you.
Thank you. Thank you. I love it if you would reach out too after.
Yeah, I can reach out to John or Corporate. Absolutely, we can connect.
Yeah, absolutely. We can connect. The same goes for anybody. If anybody needs to connect to Brenda or other of our speakers, just let us know and we'll be able to do the connection. We are past the hour, so I think we'll let Professor Ogle go. Thank you again very much, Brenda. Wonderful talk as always. Thanks for everybody being here. This was a well-attended talk. I counted, I think, 45. So thank you very much. We are excited to keep these coming. Again, the spring series are starting. And again, if you couldn't watch everything, they will be on YouTube, which Ellie actually put in the chat. You can just access them through ATP-Bio website also. Thank you so much, Brenda.
Thank you. If there was anything else in the chat, I think I just saw a couple things. If you send it to me, I can answer those.
Will do.
Bye-bye.
Thank you.